Next up, we have a joint presentation from Gary Howarth from NIST and Kristen Task from Knexus Research. They'll tell us about privacy-enhancing technology testbed for genomic data.
Well, good morning, everybody. I'm happy to talk to you a little bit about sort of two components of the same project, the PETS testbed and the component of it that we're releasing soon, and then a red teaming exercise that we did sort of based off of that tool component. NIST makes me put a disclaimer. But the... Oh, thanks. So the NIST, U.S. government agency and the Department of Commerce, we are developing a PETS testbed that's trying to provide guidelines for every component of the evaluation adoption process that's required for implementing some sort of privacy-enhancing technology. And so this is going to look like a bunch of realistic or real problems, benchmark public data that are non-sensitive, model solutions, and then from that we derive or we develop evaluation metrics, privacy attacks, and we're going to use these things to derive guidelines that help organizations to deploy these models. And specifically, we have been building a privacy-preserving federated learning system that we're trying to make easy for the community to use as a benchmark system to quickly run experiments and different evaluations. I'm not going to go into detail about it because I know our time is short. So this is really an invitation that if you're interested in using some tools, then get in touch with me. And then we can talk a little bit about this, and I'll share out the slides so you can sort of have a sense of what we're doing. But in short, what the PPFL testbed is trying to do is we have a couple different genomics data sets that are non-sensitive but real. So we have like soybeans and with actual phenotype data and lots and lots of individuals. And we also have some eukaryote data through dogs with cancer, sad but interesting. And then we take those data and then we can make quite interesting partitions that you might see, like that because genomic silos tend to have interesting distribution. So we simulate some of those. Then we have this nice federated learning system that has several different models. It's modular. And we have an easy to configure system to batch experiments, to queue things, to be able to run a bunch of different experiments relatively easily. And then we have a standardized way of producing reports with evaluation metrics. We conduct some privacy attacks. And so you can easily say, hey, I want to run, with this many clients. Here's an example of like the configuration file. So you can say, oh, I want to simulate 10 clients with, this like series of epsilon values, run the experiment overnight. And then in the morning, depending on the type of machine you're using, then you'll see like, oh, nice reports that show how do these systems compare. So I'm now going to turn this over to Christine Task, who will tell us about the red teaming that we did on these data.
Hello. And this is going to dovetail incredibly nicely with the previous talk. So, all right. First of all, we've all been talking about this, but just to make it nice and simple, differential privacy operates best with data that's skinny and tall, right? We want low dimensionality and we want a lot of people. When we're looking at genetic data for humans in real actual clinical usage, we're talking about 30 people or 50 people or 100 people. So it's short, and oh golly, is it wide. So millions of columns. And even better than that, the data is also not that interesting. It has low entropy. And this is really important to understand when we're trying to use things that we would normally apply to tabular data to this data format, which is, so you're in this room, probably you've seen something like this before, but this is the default value for the gene, and that's the value they actually have, and the vast majority is going to match the public default. So when you go to do a red teaming exercise, the first thing that we wanted to ask ourselves is, what is a meaningful reconstruction attack against something that everybody already mostly knows? Against something where the vast majority of the information in there is actually public. So we did this red teaming exercise. Fun thing about that is a batch of people trying to hack a batch of models under a set of circumstances. We learned things from designing the models and the problem to make it realistic. And we learned things from watching people hack away at it, people and two AIs. Okay, so to come back to what is a meaningful reconstruction attack? We wanted this idea of, okay, so especially because we followed common practice for doing this, and this was not a language model, probably should have been. This was just a standard classification neural network model, CNN, because that is what the literature and the genomics research was doing. But when we went to go do that, we had to look at this idea of what does it mean to be able to reconstruct something that's interesting in the gene. So we looked for individually identifying genes. Of all the millions of genes out there, you can go ahead and reduce that and say, let's just look at the ones that are high variance, high entropy. So what are the ones that actually change? Then we clustered those together, right? So makes sense that genes will be correlated. You know, this set of genes indicates some trait. That set of genes indicates some trait. So if I know one of your genes in that set, I can probably guess the next one because it's an indicator that you have that trait. So once we put those into clusters, then we can look at independence between them. And now I can say, okay, genes from cluster 1, which are independent from genes from cluster 2, which are independent from genes from cluster 3. This is the interesting meat of the data. And now I'm down to like 30 genes, and I can identify 80% of the data, individually out of 1 million people. So those are the ones that are interesting. When you're reconstructing the data, if you get those right, then I know you've actually gotten something that's interesting. We also looked at membership attack. When we did that, we did it in a realistic setting. Of course, everything here is federated learning is the idea, right? Because we said, we don't have 1,000 people at one site. We've got 30 and 50 and 100. So we're trying to do this in the federated learning setting. We looked at a realistic attack against federated learning. And so the idea there is that you have some set of records you bought from a data broker, DNA records that you know are definitely not in the data. And then you have what we're calling relevant records, which includes some things that are in the data and also some things that are not. And that's what you're using to train your attack model. So you don't have perfect knowledge of the data to try to attack the data. Okay. In this realistic setting, we also looked at subpopulation. So in addition to saying, okay, I have played with genetics using sort of standard data science techniques and said these are interesting genes because I can use them to uniquely identify people. We also can say, here is a hidden property. So here's somebody has some property that you can predict from their genetics that we know the ground truth on, and we want to know if any of those people with that property are in this data. So in this case, we're working with the dog data. Some of them were hounds. So the question is, can you reconstruct the data well enough to infer whether there was a hound in a gradient? Hound hunt. Out of curiosity, how many people here have worked on privacy and maybe privacy attacks for genetic data? Cool. How many people think that you're going to be able to do that? Find the hounds? Cool. Good. All right. Can't find the hounds. Spoiler. Okay. So we now also had to go deal with the fact that we had millions of columns of data. We wanted to do as close as we could to what the recommended practice was in the people doing classification on genetic data in federated learning, in health contexts, no thinking of privacy. So the first thing we did is we reduced the genetic data. For the soybeans, we followed the paper we were using. For the dog data, we worked with the NIST genetics folks. That's a mix of just dropping every other gene and also doing some analysis to say, okay, what are the genes correlated with the task the model's trying to classify? We also did one-hot encoding because that's what they did. How many people are familiar with one-hot encoding? Right. One-hot encoding is awful because we started out with millions, we reduced it some, and then we increased it by a factor of 4 by changing all of these, categorical things to binary values with deterministic correlations between the values. Okay. Then we created realistic client subpopulations. So it is not realistic in a federated learning setting to say that each of your client's datas are going to be a perfect exact, IID uniform random subsample of the base data set. They're going to be biased a little bit, slightly different data in different clients. And so, and that makes a big difference, as the last talk showed, right? Because when you have, one demographic at one place and a different demographic at a different place, that, substantially affects the distribution of the data, and that's going to affect how well your learning goes even before we do privacy. So in order to recreate that, because we don't have demographics on soybeans, we use the 5th and 6th PCA axis to do sort of very subtle clustering and got a subtle bias heterogeneity into our clients. We also ran from like small clients, 50 up to 200 larger, talking with health folks, and that was about what they estimated. Unfortunately, we're not getting 1,000 on anything that I've been talking to. Okay, so minimum task accuracy. This was the other thing. So there's a classification task you're trying to do. You're trying to predict the weight of the soybean oil, or you're trying to predict the breed of the dog, or something like that. We required there be at least 70% accuracy. So we never set epsilon so low that we couldn't get at least 70% accuracy. You think about if somebody is going to make a health data policy based on something they've learned that's going to affect your treatment, and it was only accurate for 70% of the population, it's not super reassuring to begin with. So we set that minimum bar because anything less, anything worse than that is now sort of a bespoke random number generator. It's not privacy preserving machine learning because we're not learning. So unfortunately, that meant that on the soybean data, which was less heterogeneous, so easier to learn, we could get down to an epsilon of 5. But on the dog data, which was much more heterogeneous, much harder to learn, thank you for that talk, 20 was as small as we could get. Okay. So I've already talked about this a little bit, but I'm going to mention it again real quick. We weren't getting exceptional performance on the task models even before we added noise for privacy. And we did everything we could to try to get reasonable performance. I expect language models are the way to go. We were using the CNNs because that's what people were doing. We went and talked to the people afterwards, and it's not clear. We could not find an example. And this we were like NIH folks. We've talked to Canadian health folks. Have not yet turned up a use case where somebody actually used federated learning on genomics data to learn new information about a medical condition that impacted clinical recommendations on practice. We've seen federated learning work out successfully, apparently, for image data like radiology stuff, but it's not on genomics. If anybody has a use case, would love to hear about it, but that's exciting to know. Something else you guys may have noticed in your own work, which is that epsilon decreased. We lost utility before we gained privacy. So the first thing that would happen is that the models would start turning out sort of random stuff, real poor utility, but they'd still be vulnerable to MIA attacks. That was fun, because I always thought when you had no utility, certainly you were just completely random, but no. But also, we did still have some privacy, so meaningful reconstruction, those interesting genes or the hound hunt, turns out to be really hard. So you could get an MIA, but the other things were not seen as successful. All right, so here's the actual results. So on the soybean data, although epsilon 10, 200, and infinity had very different levels of utility on the task, where 10 had fairly poor utility, in fact, actually, the privacy against membership attacks was basically the same. So epsilon of 20 didn't provide any meaningful privacy. And the most successful attack against the most vulnerable client model was able to get an 80% success rate. About 80%, you start to think that is actually a membership attack, that if I was worried about membership, I might be worried about. On average, you can see the success was more like 40 and 30%, which is not that bad. Like if I was a data broker saying, I'm going to sell you this list of people I've hacked into this thing, there's a 30% chance they're in here, you probably wouldn't buy. Looking at reconstruction, that was a much lower accuracy, 10% for individually identifying genes. This is important because it was 10% accuracy for individually identifying genes, but it was 60% accuracy if we just gave them credit for guessing that everything was 0. So because we used one-hot encoding, and because so much of it was zeros, if we just literally took them at their word for reconstruction and said, how much does it match? It looks like it's accurate, but you're not really learning anything interesting. Okay. We did this again with the dog data, and this time we had two new participants, Elon from MITRE and Claude. We gave Claude our competitor's pack, everything, no further instructions, just let it one-shot it. And what it figured out was that, you need to do post-processing and things like this to maintain the one-hot encoding constraints. And that was able to get it from that 10% sort of accuracy up to 40%. Elon did that in something much more clever and valuable, and this goes into our lessons learned, which is he did dimension reduction. So in order to do the attack, rather than training the attack model in the one-hot encode space, reduce the dimensionality of the data, train the attack there. Turns out that works a lot better than working in giant wide data. Like I said earlier, you can't find hounds. That was never really much better than a 50-50 shot. Okay, so lessons from this. It's a lesson from the rest of the conference too, I think. Things work better if the data is represented in a space that helps us maintain the signal as we add noise. Because, the fact that we have problems adding privacy here, it fits in neatly with the fact that we have problems maintaining the utility even before we did the privacy. If we add just a little bit of noise and the models were already so brittle and the learning task was already so not super viable, a little bit of noise throws it off completely. If we're able to work in a space where the learning task is more feasible, using the right models, using the right representation, not trying to feed things enormously wide, shallow data if it's possible, then we may be able to add noise and retain the signal just like we can in other differentially private contexts where that's the case. Default genomes are public data, and there's a lot of other public analysis on, that has happened that people aren't concerned about for privacy, and this may be a good thing to think about when thinking about how we could reduce dimensionality. Gary.
Well, I think that concludes. I'll also alert you, I'll put it up, in, that NIST is hosting a genomics privacy-focused workshop next week, so you're going to hear more of this if you come join us then. I'll put up the details on a slide in a moment. But that concludes. Thanks.
Thank you, Gary and Christine. Could the next speaker please come down to set up while we take a couple of questions? Yes.
Yeah, I'm just curious, do you have any ideas for what such a representation of life in space? Something that would enable you to maintain signal noise?
At this point, not substantially.
Yeah.
You can see like the private evolved, like the talk audio game at the beginning. Thank you. No, but probably there's expertise in this room to think about it. Two obvious things off the bat. One is that most dimension reductions high sensitivity if you do it naively, right? But if you're doing it on public data, that's fine. Another one is, honestly, there's a lot of genes that just aren't interesting. So something that would be worthwhile is which genes do I actually need for the health analysis, for the benefit, right? For the thing I actually need to learn, for the actual use case. Can I go drop a whole batch of stuff that's not relevant to it? There's the first two guesses, but I suspect people are going to think of better things. Also, Gary, the people coming to the talk at NIST are mostly not privacy folks. It's a lot of genetics folks too, right?
Yeah, I think so. Yeah,
So
it's mostly geneticists that will probably get it.
So I suspect they know things too. Okay.
Yeah.
Just one more question.
Okay, thank you. So going back to what to protect, given the low entropy of the gene data, what do you think is like a reasonable minimum unit of protection, both at the sequence level or at the higher level?
Yeah, that's a great question, and I'd love to hear some recommendations about this. And I think that maybe there's I think that there's real work that needs to be done here, and it might be like a good opportunity for like a breakout workshop. Like, what is the unit of privacy for like talking about protecting data?
It's got to go back to the representation because, like, we did it at the individual level, right? But like when we found these genes that were interesting and meaningful, they're buried in the giant pile of genes. And so I can't tell you, if you say, okay, it's a string of, we'll cover all strings of 5, for all I know, this is a super powerful string of like 20 that's right next to each other or something. So the thing I would say is the biggest takeaway is we need to understand the use for this data, like where it actually will impact health incomes, so we can work our way backwards to say, what information do you need? And then we can, I think the rest will make more sense from there. So next Thursday, right?
Yes, that's right.
Okay, next Thursday.
Let's thank the speakers again.